I think it's the problem with the projector. Oh.
Welcome everyone to the first session of this course as scheduled. I request Dr. Viprodhar sir to please begin the session. Sir, please.
Yeah, thank you. Everyone can hear me okay? Okay, great. So just give me a second. All right, good morning everyone. Can we be a little more loud? Okay, that's the spirit. So we are going to start our first session or first lecture, Introduction to Environmental Bioprocesses. Okay, so you know, like I know that it's going to be recorded, but I think you can, you know, like if you have a question, please feel free to stop me anytime and ask question. And I know that you are from very different background, right? So I'll try to provide, you know, like basic information as much as you can. And, you know, like please feel free to stop me anytime. If you don't understand any part, just ask me question and I'll try to actually answer, you know, your question. And if I can't, I'll go back, I will revisit the topic that I covered and I'll try to get back to your answer because we are going to be meeting for next 3 or 4 days, right? So, all right, so environmental bioprocesses. So we know that over 80% of the wastewater, okay, that is actually generated worldwide, it's actually dumped into surface water without any treatment. The picture for solid waste is almost the same. Okay, majority of the waste, especially in low income and medium income countries, majority of the waste that we produce actually it's dumped. Okay, so mostly we go for either open dumping or burning. Okay, so that's a common practice. You know, like I know that in many parts of India that's a common practice. Even like, you know, I'm originally from Bangladesh, we have the same thing, right? So we don't have a proper waste management system. But, you know, and because of this, you know, like without treatment, sorry, that actually I have to use proxy. So that's why I don't know why this message kind of pop up all the time. So, okay. So because, you know, like if you dump the wastewater without any treatment to river, like, you know, Professor Sharma was talking about, like, you know, coliform in Ganga river, right? So it can actually create a lot of problem. Okay, it can actually damage our water resources. It can transmit a lot of diseases, right? So it's an environmental and public health concern. And here, actually, we can use tiny microbes to solve this kind of problem. Okay, because microbes, they can actually degrade different types of toxic and organic compounds that we have in our wastewater. Okay, they can some microbes we can engineer them to actually extract bioenergy and different types of value added resources. Okay, For example, you can produce biohydrogen, biomethane using microbes. We can also produce different types of industrial chemical using fermentation process, right? So, and microbes, they also degrade plants and animals, their dead bodies, and release the nutrients to the soil, right? So, we can use microbes to solve our environmental problem, but to do that, we have to actually develop some system or develop some engineered system where actually we can have our microbes that can help us solve some specific problems. Okay. So, what is environmental bioprocesses? Environmental bioprocesses are actually the processes that utilize microbes, most particularly microbial metabolism and enzymatic activity, okay, to solve our environmental problem. Okay. So, these environmental bioprocesses can be used for our waste and wastewater treatment. Okay. We can use them for bioremediation. We can actually engineer them to produce sustainable energy or renewable energy from organic waste and wastewater. Okay. And we can also recover different types of value added resources, okay, that we can actually kind of sell in the market. So, environmental bioprocesses particularly use microbial metabolism. So, what is microbial metabolism? Microbial metabolism, it's the different types of biochemical reaction, okay, that actually occur inside the microbial cell, right? And that actually metabolic process convert the food into energy. Like we eat food, right? Why we eat food? We need energy, right? To kind of, to do our everyday activities. So, microbes, they also need the same thing, okay? They also need food, okay? So, they can degrade the food and convert that into their energy, okay? So, they can harvest energy from wide range of organic compounds, like, it could be simple sugar like glucose. It can be different other organic molecules like fat, amino acid or proteins. And like our human being, microbes also actually convert the food, okay, and conserve that energy as ATP, right? And I'm pretty sure most of you are familiar with what is ATP, adenosine triphosphate, right? Adenosine triphosphate and metabolism. Okay, thank you. Okay. So, it's all about metabolism and it's all about the energy balance, okay. Microbes, they produce energy and they use that energy for their cell maintenance, okay. And that feature of microbes can be used to design different types of environmental bioprocesses. But I want to talk a little bit more on ATP. ATP is adenosine triphosphate, right? So, it consists of adenine, ribose and 3 phosphate groups, okay? And that all energy in ATP is actually stored in this chemical bond here, okay? So, when actually microbes, they need to utilize that energy, okay? They basically just release that bond, okay? It kind of this kind of molecule, and that bond is actually kind of break and then it releases the energy that can be utilized for metabolic process, okay. So, and then from ATP we produce ADP, adenosine diphosphate, okay, is formed and energy is released to drive metabolic process. So, and as you can see here that delta G is negative. So, what delta G negative means? It's thermodynamically favorable, right? So, it can occur spontaneously. So, now going back, biochemical reactions. So, now we know that microbes, they do biochemical reaction, right? So, they can utilize organics, okay, they can degrade organics using some biochemical reaction and produce energy. So, what kind of biochemical reaction? Okay. So, that biochemical reaction microbial metabolism, that biochemical reaction actually transfer electrons from the contaminants, organic contaminants, to some electron acceptor. Okay. So if you see the glucose, okay, glucose, if microbes they use glucose, they kind of use oxygen as electron acceptor, like same as our human being. What we do for our respiration process, we take oxygen, this is electron acceptor, right, and then we release carbon dioxide, right. So here you can see that glucose is oxidized to carbon dioxide. Okay, glucose is oxidized to carbon dioxide and oxygen is reduced to water, right, and that process actually generate energy. So it's all about microbial metabolism involves actually electron transfer or oxidation reduction reaction. So what is oxidation? Release of electron, and reduction, electron taking electron, right. So here glucose is electron donor or food for microbes, and here oxygen is electron acceptor. Okay. So microbes basically they conduct oxidation reduction reaction, okay, to generate energy. So basically electron donor is transferring electron to electron acceptor, and electron donor is oxidized and electron acceptor is reduced, right. So what we know, microbes they generate energy from redox reaction, so oxidation reduction reaction, and which involve basically electron exchange, okay, between electron donor and acceptor, and one compound donate electron and other compound actually accept electrons. So microbes most of the time they use organic compound, what we have in our waste or wastewater, as electron donor, okay, electron donor, and then they can use wide variety of electron acceptor, okay, depending on what kind of process that you are using. Sometimes you know, for aerobic process it could be oxygen, sometimes for anoxic process it could be nitrate. So different kind of electron acceptor can be used, and then from this process they generate energy for their, and generate energy, and that energy is utilized for their cell maintenance and growth. Okay. And as a part of that actually, like electron, they also actually utilize for producing new cell. Okay. So part of microbial growth is actually kind of developing new cells, right. So a part of that electron is also utilized for producing new cells. So microbes they use electron donor, and then they utilize that electron donor for metabolism, and there could be two types of metabolic process. Okay, it could be either respiration or it could be fermentation. Okay, so fermentation process could be two types. It could be either aerobic or could be anaerobic. Okay, so aerobic is almost like what we do human, you know, as a human we do respiration, right. So we use oxygen, so that is actually aerobic respiration process, and that process use oxygen as terminal electron acceptor. Okay, but when you have anaerobic process or anaerobic respiration, different types of inorganic electron acceptor can be used, such as nitrate, sulfate, okay, depending on what kind of process that you are using. And but for fermentation process, organic molecules, so not oxygen or not nitrate, so in fermentation process organic molecules are used as terminal electron acceptor. But respiration process, you have to provide electron acceptor from outside. Okay, I think all lectures gonna be provided to you. Okay, so don't worry about now. Focus on what I'm telling, and we are gonna do some practice problem. But end of the lecture, it's gonna be provided to you. Okay, so yes.
Would you please put more light on this fermentation part?
Yeah. So fermentation is basically a part of metabolic process. Like larger molecules, they are break down into smaller molecules. Okay, and compared to respiration process, fermentation process produce less amount of energy. Okay, so if I give you a simple example, I don't have an paper, but I can. Is there any? Just give me a second. I need a paper. Okay. So if I am a microbe, okay, and this is my food, okay, if I do fermentation, this is fermentation, okay, but if I do respiration, it's kind of like, you know, it's a very efficient process. Okay, you can kind of completely get rid of some of the contaminants that you want. Okay, but for fermentation process, you are basically end of the process, you are going to get some organic metabolite. But obviously respiration process, I need to provide the electron acceptor from outside, right? For my respiration, I need oxygen. If you don't provide me oxygen, I'm going to die, right? The same thing going to happen to aerobic microbes. If you don't provide them oxygen, they cannot actually continue their metabolic process. So respiration process, it's much more efficient. It generates more energy. Fermentation process, it generates less amount of energy. It's not as efficient as respiration. Okay.
Organic molecule is the electron acceptor.
Yeah, electron acceptor. So for example, like, you know, from fermentation of glucose, you can get different types of alcohols or organic acid. Yeah. So those alcohols and organic acid, they are more like electron acceptor. Okay. Make sense? Okay. So let's move on. So yes, respiration process, you have to provide the electron acceptor from outside, but for fermentation process, you don't need to do that. But energy yield for respiration process, it's going to be high, but for fermentation process, it's going to be low. Yes, please.
How does it possible respiration in anaerobic condition? Can you elaborate?
Sorry?
Respiration in anaerobic condition.
Yeah, respiration you can also have in anaerobic condition. So basically, sulfate. Oh, sorry. Okay, I'll put it in my pocket. So respiration process in anaerobic condition, it's inorganic compound, it's kind of reduced to other compounds. So for example, sulfate under anaerobic condition, some microbes like sulfate reducing bacteria, they can convert sulfate to sulfide reduced form. Okay, so it's gonna be still like oxidation reduction reaction. Okay, but aerobic respiration, your product gonna be mainly carbon dioxide and water.
So does it not come under the fermentation process? That
Anaerobic fermentation process. Yes, a part of this could be, you know, respiration process. Okay, partially, but fermentation process is gonna be more dominant there. That makes sense?
Yes.
Okay, yeah. So, and anaerobic respiration, it's kind of, you know, like we are talking about here, more like soluble electron acceptor, but there are some microbes actually in nature. Like when you are going to cover microbial electrochemical technologies, there is actually topic in this course. We are going to talk about actually some microbes, they can, they don't need soluble electron acceptor. They can directly transfer electron to some conductive solid material. That's kind of a very unique bacteria in nature that can do that.
Conductive
So,
solid materials like
electrode. Yeah, so they don't need either nitrate or sulfate. So they can, instead of that, they can just directly transfer electron to some conductive surface. So that is kind of like some unique microbes. Okay. So common electron donors and acceptors, what we have in our waste and wastewater. So common electron donor most of the time is organic compounds that we have. We have different, a wide range of organic compounds in our waste and wastewater, and they can serve as electron donor. And depending on different types of like what kind of process that we are using, we could have oxygen, nitrate, sulfate, ferric iron, carbon dioxide. Often we can actually use as electron acceptor. So some examples: aerobic respiration. So aerobic activated sludge process. This is actually a picture from a wastewater treatment plant in Toronto, Canada. So in wastewater treatment plant, one of the common process is activated sludge process. Okay, aerobic activated sludge process, where you have a big tank where you have microbes. Okay, and you supply air. Okay, and those microbes use the oxygen as electron acceptor, and they break down the organic compounds that you have in your wastewater. Okay, and then they produce carbon dioxide and water. Okay, and then we can get our wastewater treated. So anaerobic respiration, one example. Okay, one example is acid mine drainage. So mining site like coal mining site or metal mining site produce a acidic outflow of wastewater. Okay, so we call it acid mine drainage. Okay, so what we have in our acid mine drainage, we have high concentration of sulfate and heavy metals. Okay, which is not that good. But sulfate reducing bacteria, so a type of bacteria that can do anaerobic respiration, so that bacteria can actually help us to solve this kind of problem. So what that bacteria do, they use like acetate or different types of organic acid that we have in our acid mine drainage. Okay, they use those organics as electron donor, and then they use sulfate as electron acceptor, and then they actually convert that sulfate to sulfide. Okay, and at the same time, acid mine drainage, we have different types of heavy metals, so that sulfide can actually react with those heavy metals and precipitate as metal sulfide. Okay, so sulfate typically sulfide is very odorous gas, right? So it kind of can also help us with heavy metals problem with high sulfate content. Okay, but the byproduct that is actually produced from this process, sulfide, can also be further converted to metal sulfide precipitate. So anaerobic respiration can actually help us actually solve this problem with acid mine drainage. Another thing, we can get fermentation. Okay, so fermentation, we have different types of microbes that we can actually utilize to convert like organics to different types of alcohol. Okay, so alcohol and organic acid, actually there is wide range of fermentation that can use. Some fermentation process can produce alcohol, some can produce long chain fatty acid, some can produce medium chain fatty acid, right? So and they have I know fermentation, first when you see alcohol you think about beer and some like other alcohols, but there are a lot of industrial chemicals that can also be produced from fermentation process. Okay, so to summarize, microbes, they generate energy from redox reaction. Okay, and they conserve that energy inside their cell as ATP. Okay, and they can utilize that energy ATP for their cell maintenance and new cell synthesis. Okay, so microbes, we can actually engineer them to actually develop different types of environmental bioprocess. Okay, to solve our environmental issues. Okay, and it mainly relies on the microbial metabolism, and that's all about redox reaction, exchange of electron, okay, and generate energy from this redox reaction and then utilize that energy for new cell synthesis and maintenance. Okay. So now let's move on. So we we already shared this equation where you have glucose that is actually oxidized to carbon dioxide, and you have oxygen as electron acceptor, which is kind of reduced to water, right? We know glucose is electron donor and oxygen is electron acceptor. But we talked about electron exchange. Where are those electrons? Okay, we don't see any electrons here, right? But we know that electrons, okay, so different compounds, they exchange electron, right? So microbes, they transfer electron from electron donor to electron acceptor, but we don't see any electron there, right? So from the equation we don't see how many electrons are transferred, but to see that, okay, so what we need to do, we need to divide that equation into two half reaction, okay. So two half reaction, so this is actually an overall reaction, but it came from two half reaction. One is for donor and one is for acceptor, right? So now we are going to learn how to establish half reaction for electron donor and electron acceptor. And you might think it's a very fundamental thing, okay, what I'm going to do by establishing some half reaction, but you are going to see later on. So everyone got calculator with you? Okay, maybe not but you can use your cell phone calculator, but we are going to actually also try to do some problem in the class. So this is kind of like my style. I try to actually also love to do some practice problem in the class, because I can share a lot of examples, okay, you are going to see and you're going to feel it's easy, and but I feel that when we do some practice problem, you maybe you are going to stuck somewhere, and at that part I can actually help you, okay, understand the concept. So we are going to learn how to build half reaction for electron donor and acceptor. So now so electron donor, we are going to use RD, reaction for donor, and for electron acceptor we are going to use RA, so reaction for acceptor. So to build half reaction, okay, so first of all we need to do we need to identify acceptor and donor, okay, and most of the cases we know that when we talk about wastewater and waste treatment process, we are going to know that organic compound is going to be the electron donor in the process, right? And then we need to know how many electron donors will be, so then we need to know electron donor will be oxidized to what, right? And electron acceptor will be reduced to what. So we have to identify that, okay? And most of the time organic compound electron donor will be oxidized to carbon dioxide, and oxygen going to be reduced to water in most cases. So let's see how to build half reaction for donor. So for half reaction for donor, we need 4 simple steps, okay. So first we have to balance the carbon with carbon dioxide. We are going to consider carbon going to be oxidized to carbon dioxide, and then we are going to balance oxygen with water, and then third we are going to balance hydrogen with proton, okay, and finally we are going to do the charge balance with electron, and at that part we are going to see actually how many electrons have been transferred, okay. So let's see, because we talked about glucose to carbon dioxide, so first let's see how to do this for glucose. So first we are going to see all carbon going to be carbon dioxide, right? So C6, we have 6 carbon. So if I have 6 carbon, so I need 6 carbon dioxide, right? 6 mole of carbon dioxide. So now you might see, okay, you know what, my reactant is glucose and carbon dioxide is my product, but why I have this on the right side, okay? So this is the actually trick for half reaction. We try to actually keep because ultimately we want actually electrons to appear on the left side of the equation, because when we are gonna build the overall reaction back, okay, so that will help. So that's why we always put that electron should always be on the left side of the half reaction. So now that has been okay. So carbon balance, so with 6 carbon we have 6 carbon dioxide, okay. So now second thing what we are going to do, we are going to do oxygen balance, okay. Oxygen balance with water. So we have here oxygen 2, right, and multiply by 6. So I have 12 oxygen on the left side, right, and on the right side I have 6. That means I have 6 oxygen missing, right. So to do that I will balance that with 6 mole of water, 6 mole of water, and then now I have how many hydrogen over there on left side? I have 12 and 6 multiplied by 2. So 12 plus 12, 24. I have 24 hydrogen on the left side. So that means I have to balance that right side with proton. So I'll put 24 proton. Now you can see you have 24 positive charges here, right? So if I need to balance, charge balance, I have to put 24 electrons there. I have to put 24 electrons there. So now I have charge balance. So I have an overall half reaction, but half reaction is always expressed as per mole of electron, okay. So whatever the mole of electron that have been transferred, I have to divide that, you know, the whole reaction, divide that by number. So here I have 24 electron. So I am gonna divide all that by 24. So I have 1/4 carbon dioxide, proton, and 1 mole of electron, and then 1 divide by 24 glucose, and then 1/4 water. So this is an oxidation half reaction. So maybe I will also use the board. If you cannot follow there, I hope you can follow from the board. So we are talking about C6 H12O6. Can you read my handwriting? Okay. So first I have 6 carbon. Anyone knew before how to do this? How to establish half reaction? Don't be shy, I am not going to be offended. Okay, yeah, so 6 carbon, right? So first we have to balance carbon with CO2. So I have 6 CO2. Now what I have to balance? Oxygen, right? So I have 6 multiplied by 2, so I have 12 oxygen, but here I have only 6, right? So I have to put 6 mole of water, right? So now I need to do hydrogen balance with proton. So I have about 12. Here I have 6 multiplied by 2. So 12 plus 12, 24, right? Hydrogen. So I am going to balance that with 24 proton. Now I have to do the charge balance. So I have 24 positive charges, right? So I have to add 24 electron, right? But now I have to actually balance everything. I have to divide everything by, because half reaction is expressed as per mole of electron, okay? So I have to divide everything by 12, okay? Yeah, so we are going to have 1 proton, 1 electron, 6 divided by 24. 1 by 24 C6H12O6. 1 by 4 H2. So that's my half reaction for oxidation or donor, right? So let's move on. So what, and now let's see another compound, acetic acid, which is a common electron donor in different biochemical processes. We can do the same thing. So we can actually do it on the board. Let's do it on the board together. So C2H4 O2, right? So how many carbon dioxide? 2 CO2. Then oxygen I have 4. So how many mole of water? 2. 2 mole of water. Now hydrogen I have 4 and 4 8, right? So 8 proton and then 8 electron, right? So I can divide both sides by 8 and I can have my half reaction. So yeah, we have 1 by 4, 1 proton, 1 electron, and then 1 by 8, and then 1 by 4 H2O. Okay, so this is an oxidation half reaction. So now let's move to reduction half reaction. Okay, so what happened to, so different types of reduction reaction that we can be interested about depending on what kind of water or wastewater process that we are dealing about. So if we talk about aerobic activated sludge process in wastewater treatment plant, we are going to be interested about oxygen to water, right? Because oxygen is electron acceptor in most of the aerobic bioprocess. But sometimes we may also need to deal with nitrate to nitrite, like for example nitrification process. Sometimes we may be interested about nitrate to nitrogen gas for denitrification process. Okay, so we need to know what would be the reduction half reaction for them. So for reduction half reaction, so first same as this thing, but we don't need to do carbon balance. We are going to start with oxygen balance to water, and then hydrogen balance to proton, and then finally we are going to do charge balance with electron. So almost same as like oxidation half reaction, but instead of 4 steps we have 3 steps for oxygen to water. So let's see. So oxygen to water, okay, so we have 2 mole of 2 oxygen, right? So we are going to have 2 mole of water. So first we are going to balance oxygen, and then, but again why it's on left side? Because we want electron on the left side of the reaction. So now we are going to do hydrogen balance with proton. So we have 4 hydrogen, so that means we have 4 proton. Okay, we have 4 positive charge, so 4 electrons. So I can actually do it on the board quickly. So we are talking about oxygen reduction to water, right? So I have 2 oxygens, so I have to put 2 water now we have 4 hydrogens, so I have to put 4 proton, and I have 4 positive charge, so that means I have to put 4 electron. And now if I divide by 4, electron plus proton plus 1 by 4 water, so that's going to be my reduction half reaction. Okay. So now yeah, I don't know, like here actually I have to use proxy for the internet, so it's kind of like showing this message every time. Okay, so this is my reduction half reaction, right? So how about nitrate to nitrite? So there, instead of like, you know, here we did oxygen balance, right? But here we have to do nitrogen balance first. Okay, nitrogen balance first, and then oxygen balance, and then hydrogen balance, and then electron balance. Okay, or charge balance. So one nitrogen. Okay, so one nitrate gonna give one nitrite, right? So we want to convert nitrate to nitrite. So now one nitrate to one nitrite, and then now we have to do the oxygen balance, right? So we put here, I have 3 oxygen, here I have 2, so I have 1 oxygen less. So what I am going to do, I am going to put 1 water molecule here. Now, so here I have 2 hydrogen. Okay, so I have to do the hydrogen balance, so I am going to put 2 proton on right side, sorry, on the left side, and then I have to do the charge balance. So charge balance, what I would do, I have 2 protons, so I'll put 2 electron. So let's do it on the board, like, you know. So I kind of like, you know, show step by step, but I think after you practice, we can just, you know, do it in one single step. So we want to do nitrate to nitrite. So NO3 minus to NO2 minus, right? Okay. All right. So first I have to do nitrogen balance. It's done. I have 1 nitrogen, so I don't have to do anything. I have 2 oxygen. Here I have 3 oxygen. So that means I have to put 1 water here. Now hydrogen. I have 2 hydrogen, but that side I don't have any hydrogen, so I have to put 2 proton. But I have 2 proton here, so there is a charge imbalance. So to balance that, I am going to put 2 electron, and we are going to do the same thing. We are going to divide. We can actually divide that by the number of electron or mole of electron. So electron, proton plus half nitrate, half mole of nitrite plus half mole of water. Okay, so that's my reduction half reaction. So that's my reduction half reaction. So I think you can simply do nitrite to nitrogen gas. We are going to do the same thing, but here it's little bit different. You can see one side we have 2 nitrogen, and other side nitrogen we have gas, so we need 2, right? So that means we either put 2 on this side or put half on the other side, right? So let's, you know, instead of seeing it on the board, maybe let's try to do it by ourselves. So we have nitrite to nitrogen gas. Okay, I think one thing I made a mistake here. Okay, so what was the error?
Charge
No, actually, like
balance.
Yeah, charge.
Nitrate.
Oh, no, actually it was correct, because here you can see there is a negative charge. Okay, so here, but now it's gonna be, like I have to put one electron less. Okay, but anyway, let's do it. So nitrate to nitrogen. So I have 2 nitrogen, here I have 1, right? So I can put a half here. So now what we are gonna do? Oxygen balance. So I have 2 oxygen, so that means I need 2 mole of water. I have 4 hydrogen, so that means I have to put 4 proton. Now how many electrons I'm gonna put? 3, right? Because I already have 1 negative charge here, right? So that's why I'll put 3 electron. So now if I divide 4 by 3, 1 by 3, 1 by 6, 2 by 3, right? So that is my reduction half reaction, right? So 1 by 6 nitrogen, 2 by 3 water. So now this is half reaction for reduction. So after we know the half reaction, so half reaction can tell us, okay, how many electrons have been transferred, and from if we can establish the half reaction, okay, for both electron acceptor and donor, we can come up with an overall reaction. Okay, we can come up with an overall reaction which is going to be the reaction for acceptor minus reaction for donor. So now establish half reaction and the overall reaction for aerobic oxidation of glucose. So again, going back to that same example, glucose and oxygen, okay, but fortunately we have everything already from previous example, so we don't need to redo them. But if I put Ra minus Rd, now already we established half reaction for donor and acceptor. So if I put Ra minus Rd, so I can have this thing, we can do some simple rearrangement. So proton we can get rid of them, electron we can get rid of them. We have half mole of water and quarter mole of water here, so we can just balance that. So and after rearrangement, and if I multiply by 24 by both sides, we are going to get exactly same equation that we had. Okay, but that equation doesn't tell us, okay, how many electrons have been exchanged, but from half reaction we can see that. Okay, but now let's kind of step back and think about why microbes they do oxidation reduction reaction. So they do oxidation reduction reaction because they want to generate energy, and at the same time they want to grow, right? So microbes, they want to generate energy, and at the same time they want to, so microbial growth means development or synthesis of new cells, right? Yeah, so they want to also produce new cells. So electron donor microbes, they are actually transferring to electron acceptor, and we already talked about like that electron transfer process, right? And that electron transfer process actually generate energy. We have 2 half reaction, one for donor, one for acceptor, and from the difference, like we can have an overall reaction, which is that has been so. Okay, maybe for next lecture I can use a different laptop, so that would solve the problem. Okay, so so that is actually energy generating reaction, right? So Ra minus Rd. Okay, so we can call it energy generation. But we know that microbes, they also actually So we already learned about this thing, but microbes also actually from this process a portion of that electron they use for new cell synthesis. Okay, they develop new cells, and the process also requires some inorganic nutrients like nitrogen, phosphorus, iron. Okay, but they also need electron. So a portion of electron is actually also transferred to the synthesis of new cells. So far what we have learned, that doesn't include any cell synthesis. That just include the oxidation reaction for energy generation, the reaction for transfer of electron from donor to acceptor, but that doesn't include the cell synthesis part. So how to do that part? Okay, how to do the cell synthesis? So that means we need another half reaction for cell synthesis, RC. So we talked about RA for acceptor, RD for donor. So now we want to introduce another reaction called RC, okay, which is for new cell synthesis, and this is something very critical when you are talking about environmental bioprocess, because we are using microbes and we are going to know that actually part of that process, okay, also going to produce new cells, and you cannot overlook that part. For example, like let me give you an example. For example, like if you go to a wastewater treatment plant, I'm pretty sure you are going to go to visit a plant, I think tomorrow, right? And you are going to see possibly they have a actually big tank where you have bubbles coming out from the bottom, okay, that is called aeration tank, where you have microbes, okay, and that microbes actually degrade organic compound, okay, and convert that into carbon dioxide and water, and at the same time the process generate huge amount of sludge, and you have to deal with that. Yes, you are treating the organics, okay, you are treating wastewater, but at the same time you are producing a significant amount of new cell biomass that you have to deal about, right? So that you have to deal with. So we cannot actually overlook that part, okay. So far what we have learned, it's for acceptor and donor, but now we want to learn how we can incorporate microbial synthesis, like in this equation. So we are going to introduce a new reaction, which is going to be RC minus RD, because here donor reaction going to be the same, right, half reaction. So we need another half reaction for cell synthesis, okay, and that part we are going to learn. So now if you look into the overall picture, we have donor, we have electron acceptor, we have new cells, okay. So and we have three half reaction, one is for donor, one is for acceptor, one is for cell synthesis. So for that part we are going to assume that, okay, a portion of the substrate electron or electron donor going to be utilized for energy generation, and we call it this Fe, or fraction of electron utilized for energy generation, Fe, and we are going to introduce another term Fs. So portion of electron that is utilized for new cell synthesis, okay. So and Fe plus Fs, if you consider one electron, then Fe plus Fs going to be equal to 1, okay. So overall reaction going to be Fe multiplied by Re, energy reaction, and plus Fs, fraction of electron utilized for cell synthesis, multiplied by Rs, right? And we already know how to establish this reaction. Now we are going to know how to establish this reaction and how we can get an overall reaction from here. So now if I like Re is Ra minus Rd and Rs is Rc minus Rd, right? So if we do a little bit of rearrangement, what we get, and we know that Fs plus Fe gonna be equal to 1. So we get overall reaction Fe multiplied by Ra. So fraction of electron utilized for energy generation multiplied by acceptor reaction, and then Fs, which is basically fraction of energy utilized, electron utilized for cell synthesis multiplied by RC minus donor reaction. Okay. And to give you an example, just a moment ago I was giving that example that in wastewater treatment plant, in aerobic activated sludge process, actually major portion, okay, only 60% is actually going to acceptor and 40% is going to new cell synthesis, and that's why you see a significant amount of sludge production from aerobic processes, okay, that we have to deal with. But compared to, so that means that Fs is going to be 0.4 and Fe going to be 0.6. Okay. So, major portion of substrate electron is actually going to new cell synthesis, but this Fs for anaerobic process is much more smaller. Okay. So, anaerobic process typically it's 5 to 10%. Okay. Maximum it could be 20%, but not more than that. So, that is why aerobic process produce more sludge than anaerobic process. Okay.
Could you please explain this point again? I could not.
Yeah. So, here, so what we are saying, so donor is basically providing electron for microbial metabolism. So, what we are saying in aerobic process, a majority of the electron, okay, yes, still majority 60% of the electron going to go to the terminal electron acceptor. So, it is going to react with oxygen, okay, but still a significant portion like 40% of that electron going to go to the new cell. So, 40% portion of the organic going to be utilized for new cell synthesis. So, microbial cell, what we have? We have carbon dioxide, hydrogen, oxygen, right? So, that thing going to come from the organic. Okay, microbes, they are going to utilize those organic to produce new cell. That makes sense now? Okay, thank you. All right. So, now how we can develop half reaction for cell synthesis? Okay. So, for microbes to develop new cells, they need carbon, they need hydrogen, they need oxygen, but at the same time they also need nitrogen, right? Because cell are protein, and protein you need nitrogen, right? So, microbes, they can use different types of nitrogen source, like they can use ammonium, they can use nitrate, they can use nitrite, they can use nitrogen. Okay. So, a wide range of nitrogen can be used for new cell synthesis. So, how to establish the half reaction for that? And so, thanks to many researchers actually who already developed this kind of cell synthesis reaction, half reaction. Okay. So, so this is half reaction for when actually ammonium is utilized for new cell synthesis, and this is for nitrate, nitrite, nitrogen gas. So, when we kind of work with this, okay, so to establish it was not very easy, like it was actually done experimentally and then validated. Okay, but we can actually directly use those cell synthesis half reaction. Okay, but one thing you are going to notice here. So, where is cell here? Can you show me?
Carbon, nitrogen, oxygen, nitrogen part.
So, C5H7O2N is the actually microbial cell. This is an empirical formula for microbial cells. So, how researcher came up with any thought how they came up with this kind of chemical formula?
They could have taken all sort of microbes present in wastewater, and then they could have analyzed this CHON part.
Yeah, exactly. So, if you know the elemental content, okay, even if I know my elemental content, I can have a chemical formula for myself. Okay. So, researcher what they did, they got the biomass, okay, that is actually produced in the process, okay, in wastewater process. and they did elemental analysis. Okay.
I have a question. Is this only for activated sludge process or for?
Mostly for aerobic biomass, I would say, aerobic heterotrophic biomass.
Okay.
Yes.
So activated sludge process.
Yeah, aerobic heterotroph. Yeah. So most of the cases we use this empirical formula C5H7O2N. Okay. So now let's go back. Now we are going back to same glucose and oxygen, same example, but now we are saying that we are going to have a cell synthesis reaction and nitrate going to be the nitrogen source. So that means we have to use this half reaction. Okay, this half reaction using nitrate, and we are saying that Fe, energy utilized for fraction of electron utilized for energy generation is 0.6 and cell synthesis is 0.4, just like the example that I was giving. So fortunately we don't need to establish any new half reaction here. We have all of them from different slide, example 1, example 3, and from slide 34, that my previous slide. So now this is my reaction, overall reaction, right? So first what I have to do, acceptor reaction I have to multiply by Fe, which is 0.6. So I have the acceptor reaction, so I multiply by 0.6 and I have this. Now cell synthesis reaction I have to multiply by Fs, which is 0.4. So I multiply by 0.4 and I have this reaction, and Rd I also have. So my overall reaction gonna be this. So this is my cell synthesis reaction. Okay. So this is my overall reaction considering my, including my oxidation, reduction and cell synthesis reaction. So basically representing both energy generation and cell synthesis. So overall reaction. So how that process, like we have an overall reaction now including cell synthesis. How we can use this equation? Any thought? What could be the practical application of this equation?
Shall I say?
Yeah, sure.
Because mostly they are students, so I thought.
Yeah.
So actually in calculating how much is the load in wastewater, and so basically it's about F by M ratio, food by microbe ratio.
Exactly, yeah.
So we have to know how much nutrient is there, and then we can grow the microbes accordingly.
Yeah. One important thing, for example, we can say, okay, you know what, how much sludge gonna be generated from the process? We can estimate that using this equation, right? Or if I wanna consider, okay, how much oxygen I have to provide in the process, right? I can get that from this equation. So lot of things, yes, it's kind of a theoretical equation, but we can still actually kind of use that equation for practical implication. Okay, and environmental bioprocess is all about that. It's this morning I was telling, if you learn one technology kind of in detail, like you can learn other techniques like very easily, and that's the beauty of environmental biotechnology. Okay. So now going back, I started with saying that okay, 80% of the domestic sewage is actually dumped in surface water without any treatment, right? And we know that microbes work. Okay. Now why we are still actually dumping wastewater without any treatment? And this is actually, I got this this morning from I was searching online to see some statistics from India, and this was from Times of India in 2021. You can see almost more than 50% of the wastewater in India, it's still actually dumped to the river or other surface water bodies without any treatment. And why is that? We know that actually the we can infrastructure and technical support.
But mostly from my practical experience, major issue is the operation and maintenance problem, other than the infrastructure nowadays.
Yeah.
Because
So
governments create the facility, but they cannot check operator is really operating or not.
Yeah, and that's kind of like, most of the time out of sight, out of mind, right? Like you flush your toilet, everything is gone, and then this is not something fancy thing to talk about, right? So so that's actually an infrastructure. Biological process, one of the major limitation of biological process, it's a slow process, right? So that means you need a very large footprint of bioreactor to treat. And think about population, right, in India or in Bangladesh or some other like countries. Like you are dealing with a significant huge volume of wastewater, and it's not actually we know the fundamentals of the process, but the challenge is how we can make it cost effective, how we can actually make the microbial process faster, right? So we have a lot of things to do. Okay, yes, like there are a lot of textbook there, we know the fundamentals, but we have to do a lot of things in the field. So, and that's actually the that's why actually this topic is really important. Okay, yes, sometimes like it's not as fancy as other topics, but still it's something very important we cannot overlook. Okay, so now let's get into business. I want to do few practice problem to see like whether you could follow what I was telling. Okay, so let's start with maybe we can spend 15 or 20 minutes. I have 3 practice problem. Okay, but if you stuck anywhere, please let me know. I'm here to help, and that's the main idea about like about this course. Okay, so if you stuck anywhere, just let me know. So first we are going to build 2 half reaction. Okay, you can see 2 organic compound, one is pyruvate and one is benzoic acid, and let's establish the formula there. So why don't we spend maybe, I don't think you need a huge lot of time for this problem, but let's see, we spend about 5 minutes for this. I put a timer on my cell phone so we can track the time.
Continuous 2 hours. So if you want, you can take 5 minutes break also.
I was thinking I need another 20 minutes to finish this module, and then we'll give a break.
Okay.
And then I should continue now or after lunch hour.
We have up to 1:30, but you can maybe you can extend up to another, and then
I can.
So this is up to 130 and then 4 to 530. So you can continue in that also.
Okay, okay.
Maybe.
Okay, I can continue and then you can go for the last module today.
Yeah, I can do that.
Okay, yeah.
Maybe after lunch you can continue.
Yeah, I'll
at the
continue.
end.
Okay, yeah, sure. Done? Okay. Yeah, I can come and have a look. Yes, sit there.
Is it correct, sir?
Okay, so you have 3 carbon here, right? So you need 3 CO2, right? CH3COCO. So that means 3 carbon.
Yes.
So now you have to balance it again. But
Six
what are
oxygen.
gonna be on the other side?
3 oxygen already here.
Okay.
CH2. Balance oxygen.
Yeah. Now you have to.
Please explain for one, sir. Another will use.
Okay, yeah, sure. Okay, so what I would do, I would explain one, okay, and then I'll leave the second one for you. But I'm pretty sure some of you could do that already. So maybe I'll use the board, okay? I think it's a board is better to explain. So we have pyruvate, CH3 COCO minus. So what first you should do? Carbon to carbon dioxide. So how many carbon we have? 3. So that means I have to put 3 CO2. Now oxygen balance. I have 6 oxygen on this side. So I have 3 over there, right? So how many water? 3. Now hydrogen balance. 6 plus 3. I have 9. Right? So how many proton? 9 proton. Now charge balance. So charge balance, I have 9 proton, but if I put 9 electron, the problem is here you see there is 1 negative charge, right? So I have to put 1 extra to balance, right? So that means I would put 10 electron. Okay? 10 electron. So then I divide per mole of electron 9 by 10, 3 by 10, sorry, 10, 3 by 10 H2. Make sense? So look into the charge, okay, and like I think some of you might wrote 1 mole of CO2, maybe just look into the first thing here, but you have actually 3 carbon, right, on this side. So now how about benzoic acid, second one? Huh? 6 by 26, okay. Oh, like, am I missing something for the second one? Do you want me to do it on the board? Okay. So second one, I can share the answer. So I have 7 by 30 CO2 plus 1 proton, 1 electron, 1 by 30 H5COOH plus 6 divided by 15. Everyone got this? Use the config. Actually, when I use the proxy, it always shows that message. Yeah, because it's kind of need the internet connection, so that's why it's always showing that message. Maybe for next class I will use. Yeah. Okay. All right. So should I move to the next practice problem then? Next practice problem is actually one of my favorite. Okay, it's kind of very interesting one. You can see some application. A wastewater treatment plant has decided to use biological denitrification process. So basically nitrate to nitrogen gas, which is actually part of nutrient removal. We call it biological nutrient removal process because if you don't remove nitrogen and phosphorus from the wastewater, you can have eutrophication. So basically you see algae bloom in summer, right, in water. So those blue green algae. So here in this process, so denitrifying bacteria, they need electron donor. Okay, you have to provide electron donor from outside. And most of the treatment plant, they provide like different types of organic acid or alcohol like ethanol. Okay, so here in this example we are saying ethanol will be used as an electron donor for denitrifying bacteria. Calculate the annual cost of ethanol required for 90% nitrate removal. Okay, so we want to remove 90% of the nitrate, okay, from the process, and we want to estimate how much money we have to spend for that. Okay, like that's a like this is another way you can see like how those half reaction can be utilized for practical implication. And wastewater flow rate is 80,000 meter cube per day. Nitrate concentration is 60 milligram per liter, and price of ethanol is $0.15 per kilogram, and assume FS is negligible. So what means FS is negligible? FS is negligible means we are ignoring the sludge production part. Okay, so to simplify our life. Okay, so we don't need to. So we only need to consider the RE, reaction for energy generation, acceptor minus donor. And I know you can. So first, I'm giving you some hints. Okay, and maybe give you 5 to 10 minutes. And then we will go through the solution, but please try to solve it by yourself first. Okay, so what we need to do first, we need to establish a half reaction for donor. So what gonna be the donor here? Ethanol, right? So for ethanol we are gonna establish a half reaction. And what would be the acceptor? Right, that nitrate to nitrogen, this is going to be my acceptor reaction. So we are going to complete this reaction, and then we are going to have RA minus RD, overall reaction, RA minus RD, right? And then based on that, from the final reaction we can tell, okay, you know what, like for per mole of nitrate, how many mole of ethanol I need, right? So based on reaction stoichiometry, I can calculate how much ethanol I am going to need for 90% nitrate removal, and I know the price, I can calculate what should be my cost for this process. Okay, so let's try it. Okay, if you stuck somewhere, please let me know, but I am going to show the solution. Okay. But at least try to establish the half reaction as a starting point. So if you have RA and RD, you can calculate the overall reaction. So let's try that. Anyone could get the overall reaction? Are we done with that part or not yet? Can I move on? Okay, I'll give few more minutes. So this is my overall reaction that I got. I can quickly write it on the board. Anyway, you have half reaction there. So overall reaction I got is 1/5 nitrate plus 1/12 C2H5OH plus I think 1 mole of proton, 1/5 proton, right? Then 1/10 nitrogen, 7/20 water plus 1/6 CO2. Okay, so this is my overall reaction. But for this problem, honestly speaking, I don't need to need the whole thing. What I really need, I need to know how many mole of nitrate and how many mole of ethanol I have, right? So I can see 1/5, which is 0.2, right? So 0.2 mole of nitrate. If you want to convert, then I need 1/12, which is 0.83 mole of C2H5OH, right? So 0.2 mole of nitrate, if you want to convert to nitrogen gas, then you need 0.083 mole of ethanol, right? Now I know the molecular weight of nitrate. Nitrogen is 14 plus oxygen 16 multiplied by 3, 48 plus 14, about 62, right? So nitrate is molecular weight 62 gram per mole. How about ethanol? Ethanol is 12, 24, 24, 5. 529 plus oxygen 16 plus 1, 46 gram per mole of ethanol C2H5OH, right? So now that means 0.2 mole, right? So 62 multiplied by 0.2. Everyone can see my writing from the back? Okay. 62 multiplied by 0.2. So 12.4 gram of nitrate. If I wanna actually convert, right, what I need? 46 multiplied by 0.0833 something gram of ethanol C2H5OH. So 46 multiplied by 0.0833, 3.83 gram of ethanol, right? So this is what I need. So 12.4 gram of nitrate, 3.83 gram of ethanol. Now let's go back to the question part. So now I want to remove 90% of nitrate. So now I have to go back to the question. I have to see what is my nitrate loading in the process. Okay, what is the total kilogram of nitrate is actually entering to the system, right? So then I can actually use this stoichiometry to actually calculate how much ethanol I need. So to do that, I have 80,000 meter cube per day, right, is my flow rate. Concentration is 60 milligram per liter. If you convert, that's going to be 0.06 kilogram per meter cube, right? And I want to get rid of 90%, right? So I'm not looking for 100%. I want to remove 90% of the nitrate. So my nitrate loading in the process is going to be so 80,000, right? 80,000. 80,000, okay. 80,000, okay. 80,000 meter cube per day, and I have 0.06 kilogram per meter cube, right, and per day, and I want to estimate annual cost, right? So 365 days. Sorry, yeah, 90%. What I will do, I'll just multiply by 0.9, right? I need 90% removal. So my loading, I'm going to consider it's like 0.9, and it's for 1 day, so I can multiply by 365. So what would be my loading then? 0.9 multiply by 80,000, 0.06 multiply by 365. I have about 155. Did everyone got the same number? No? Yeah, one. I'm getting a different number now. Why? Okay. So 0.9, 80,000 multiplied by 0.06 and then 365 days per year, right? So 0.9, 80,000, 0.06, 365 days. Now I'm getting 157,6,800. Are you getting the same? Okay. So now I can go back. So now I know what is my nitrate loading, right? So that is actually my nitrate. Now from the half reaction, we got a stoichiometry that 12.4 gram of nitrate. If you want to convert, right, to nitrogen gas, you need 3.83 gram of ethanol, right? So now I can use that. So 12.4 gram of nitrate. We need 3.83 gram of C2H5OH. But I can write both side kilogram and kilogram, right? It's gonna be same. I can write kilogram and I can write kilogram, right? So now I have now 157,6,800. okay, kilogram of nitrate. So that means 3.83 divided by 12.4 multiply by 1576800, right? So 3.83 divided by 12.4. So we need about 487027.7 kilogram of ethanol. And each kilogram is about, what is the price? 0.15, right? So if I multiply by 0.15, it's about 73,000. Okay, $73,000 that I need. So if I multiply that by 0.15, I need about $73,054. Okay, if you convert that to Indian rupee, $80 in USD, so almost 500,000 Indian rupee. Okay, so you need annual cost. So this is here I got about like I think 73,000. Okay, yeah, exact same number I got there. Okay. So yeah, so this is kind of like an example how we can use the half reaction to, even you can do cost assessment for the process using half reaction. Okay, so I have a very last practice problem. Okay, but all I think this lecture gonna be actually shared with them, right? So after the like after the thing is done. Okay, so very last problem, but is it possible, because they are going to have a quiz, is it possible if we can share them at the end of the day, we can share the slide, so you can kind of review the topic and get you prepared for the quiz. Okay.
I think it's the best idea. After every day.
Yeah. So yeah, at the end of the day we can share the slide. Okay. And where you are going to have everything, so you can actually prepare for your quiz on Friday, right? And that's when all the magic going to happen. So, but that quiz is going to be mainly MCQ type, right? So MCQ type, so you don't have to do the calculation there, but still I feel that this is a very good practice. It's going to be something I hope useful, okay, for you. So anyway, very last practice problem before we take a break. Okay, the representative empirical formula of biodegradable organics in a wastewater sample is C8H17O3N. Let's assume that the oxidation of biodegradable organics in a biological process can be expressed with the following half reaction. So you have a half reaction there. It's already we built. So yeah, that was actually so you already have a half reaction there. Okay, for the oxidation, so you have the half reaction for RD, right? And the released electron from oxidation process can be used to reduce carbon dioxide to methane gas in a bioprocess. So it's kind of like a, anaerobic digestion process, which is an anaerobic bioprocess where actually carbon dioxide can be reduced to methane gas. So this is a reduction reaction, right? So here you are going to have a half reaction for RA, right? So RA. So now you have to establish an overall reaction for the bioprocess, and your Fs, which is fraction of electron utilized for cell synthesis, is 0.08. okay, 0.08. So, and they use ammonia as a nitrogen source for cell synthesis. Okay. So, we want to actually establish a overall formula, overall chemical reaction. Okay. But I know that you don't have all the half reaction handy, so I'm going to go through this example. Okay. So, but again, I would recommend end of the day when you have the lecture, please go through the materials. Okay. Try to practice those half reaction by yourself. Okay. And it's kind of quite useful, and it's a good practice as well. So we have this half reaction was given in the problem, and we already know those half reaction, but maybe for the methane part we can do it quickly on the board, right? Because that's a new reaction, we didn't do it before. All right, so we want to do half reaction for carbon dioxide to methane, right? So carbon dioxide to methane, we have one carbon, so we don't need to do anything, right? We have 2 oxygen, so that means I have to put 2 mole of water. So now here I have 8 hydrogen, so that means I have to put 8 proton. And to balance I need 8 electron, right? So now if I divide by 8 on both sides, 1 by 8 CO2, 1 by 8 methane, 1 by 4 water, right? Yeah, that's exactly what we got. And now the third part, RC we have a list, right, in the lecture note. You can also find that in environmental biotechnology book. Not every environmental biotechnology book actually include those half reaction, but one actually written by I think Perry McCartney and Bruce Reitman. Okay, there is an environmental biotechnology book you are going to find there. But anyway, I have that list in the lecture note. So then Fe is basically fraction of electron utilized for energy generation. Here we said fraction of electron utilized for cell synthesis is 0.08, right? Only 8% of the electron is utilized for cell synthesis, and this is common because it's an anaerobic process, right? So 92% electron gonna go for energy generation. So this is 0.92. We are gonna accept a reaction. We are gonna multiply, and Fs, which is 0.08, we are gonna multiply the cell synthesis reaction, RC. And then we have this equation, okay, where we can actually plug all those numbers, and we can try to find this overall reaction. Okay, so I would recommend. I did not show the full step here. Okay, only wrote the final, but you should try that at home. Okay, so please try that. And so here we end the first session. Okay, we will take a break, and then. So how long we should take a break? Oh, we are going to actually stop now?
No, if you want to continue, then another 10.
So it's 1:10.
Yes.
So I
You want to
think
finish this one?
I'm done with this one.
Okay.
That was the last slide for this session. So why don't we continue at 2:30 then?
Yes, because I think you need more time than 15-20 minutes.
Yeah, maybe yeah, I'm okay with 2:30. So what's in the schedule?
Stretch the second half.
Okay, yeah. So in second module we are going to cover So now we want to jump into wastewater treatment. So far we talked about the basics of environmental bioprocess. Okay, so we learned how to establish half reaction. We learned actually how to actually what is the practical implication of half reaction. Now in second module today we are going to talk about characteristics of wastewater. Okay, so more like talking about COD, BOD. Okay, some of the implication. Okay, and we are going to learn about wastewater treatment process, and tomorrow we are going to actually spend more time on kinetics. Okay, microbial kinetics, because why kinetics is important, because as I was telling that microbial kinetics, it's slow process, right? So that's why you need a very large footprint of your system, which is sometimes not very cost effective to build, right? So everything all the design equation or process parameter, they are kind of linked to the microbial kinetics. So tomorrow morning, so today we are going to learn wastewater characteristics. Tomorrow morning we are going to learn microbial kinetics and little bit more detail on wastewater treatment process. And then tomorrow a major portion of our time we are going to focus on how we can use kinetics. Okay, we can actually kind of translate those kinetics into some design equation, and how we can actually utilize this design equation for talk about different types of important operating parameter or design parameter for wastewater treatment system. Okay, so that's the whole idea that we have. Okay, so thank you so much, and see you at 2:30 then. Okay.